Hi everyone, thank you for joining the webinar on transitioning to animal-free cell culture media in India. My name is Dr. Ankita Pandey. I'm an advisor to PETA India and I'll be moderating today's webinar. We are glad that you all could join today. This webinar is particularly timely, aligning with the Indian Pharmacopoeia Commission's recent draft on General Chapter 2.7.2, Cell Substrates for the Production of Vaccines for Human Use, that was released for stakeholder comments. In the draft, the Indian Pharmacopoeia Commission has considered the information on non-animal replacements for fetal bovine serum and other animal-derived products commonly used to manufacture human vaccines. Transitioning to an animal-free cell culture media provides an opportunity to enhance the reproducibility, reliability, and scalability across several other fields as well, such as in vitro research and testing and cultivated meat production. So moving on, today we will first hear from Dr. Pratiksha Palahe from HiMedia Laboratories. Before I introduce the first speaker, I would like to point out that there will be time for questions for the speakers at the end of the presentation. Everyone is on mute, but you can type your questions or comments in the question and answer box at any time during the presentation. This toolbar should be just below your screen. Dr. Pratiksha pursued her bachelor's and master's in science and received her PhD in bioanalytical science from Mumbai University in 2016. She served as the director admin of Guru Nanak Institute of Management Studies and Business School, Mumbai from 2023 to 2024 to bring about administrative reforms, methods, and procedures for smooth functioning of the institute, developing entrepreneurship and skill division in management. She's a recipient of many awards and has several research papers and book chapters to her credit. With 10 years of heading the commercial and R&D division of National Facility for Biopharmaceuticals, Mumbai, she is working as a project lead at Cell Biology Division at HiMedia Laboratories since 2025, focusing on branding and business development. It is now my pleasure to turn the floor over to Dr. Pratiksha.
So good evening, everyone. I believe that we have people from all across the world over here. So it's evening in India. So good evening to everyone present. Thank you so much for joining in for today's webinar. To begin with, I would like to extend my thanks to Dr. Ankita Pandey for giving me an opportunity to be presenting on a very interesting topic and a topic which is very close to my heart, that is transitioning to animal-free cell culture media with prime perspective to look at what India and how India is moving in this segment. So Dr. Ankita, with your permission, I'd like to just share my presentation. Just let me know when to begin, and if you can just see my presentation, just give me a heads up.
Yeah, it looks good.
Great. So thank you so much, everyone. I think we will just begin with the presentation. I will just quickly run through to some basic information regarding this whole transitioning process into the animal-free cell culture composition. And then I would very specifically try to highlight the contribution that my company, HiMedia Laboratories, is making in this segment, which is very, very significant from the Make in India concept and pioneering the research in India on a serum-free media development. So my presentation basically would be talking about what are the different kinds of animal-derived materials that are available, FBS with specific focus on its composition, the function, how it is made, its market potential, the global demand that this FPS has in the research segment across the globe. And then how do we balance the ethical and the scientific concerns with respect to FBS? Because at one end, we have to promote science, we have to promote new technological developments, we have to look at better outcomes in terms of therapeutics and diseases. But on the other end, there's something which is very essential, that is talking on the ethics front, like how ethical it is to work on a product which is, I would say, literally cruel on the animal front. So, and then India's contribution in the global scenario for joining in with the global players, especially with the amendments which has come in the Indian Pharmacopoeia Commission, then the scientific and the regulatory challenges that we would face on transitioning from this serum-based media to a serum-free media. And are we regulatorily ready to accept these kind of changes? And then a quick run through on one of our products, which is the new Sera, which is a serum-free media developed at HiMedia. The validation data to show how this media is transitioning from a classical serum-based to a non-animal based product. And then the other product portfolios that we hold on the serum-free front or on the non-animal front specifically, conclusion of my talk and then my general acknowledgements. So if we have to look at a global data which was conducted by on a base of a survey to see whether what kinds of animal derived materials are routinely required in the scientific experimentation sphere, we see that having a serum as one of the most important, or I would say an inevitable component within the research fragment, followed by antibodies, then enzymes, growth factors, coating materials, printed materials, etc. And coming to a more pertaining or a more pertinent question, that is, even though the serum consumptions are so high, we have still a very, very big amount of risk factors which are being associated with this kind of a product being extensively used, and that is two major concerns. One is the low reproducibility and the batch to batch variability because it's coming from an animal derived source directly from an unnatural source. And the second is the ethical concerns, which is more teeming or more, I would say, depressing as compared to the low reproducibility part of it. So it was a global survey with almost 52 countries participating in this. So I think it's a very, very comprehensive and a unbiased data, which talks a lot about a singular platform of research which is being conducted all across the globe. So what pertains to one part is definitely having a lot of significance and impact on the other one as well. So coming to serum, which is one of the biggest products which has been extensively used in developments of therapeutics, or I would say technologies or antibodies or monoclonals or vaccines. You name, the fragment and serum is present over there. It basically has a very standard composition of hormones, transport proteins, growth factors, lipids, minerals, elements, detoxifying factors, each one of them significantly contributing in some or the other way to the activity. So having such a vast composition and having such a great impact on not only the growth of the cells, but also on the viability, on the metabolism, on the proliferation, on all kinds of activities within the cell. Serum plays a very, very significant role in the research in the biopharma, or I could say in the cell biology fragment. So coming to the FBS, which is present on your research table, how does it reach your table? It's a very gruesome and a very cruel method of uh, commercially having FBS on your table. And that method is basically like cutting the calves from unborn calves from their mothers, at the fetal stage, and then puncturing their heart and using a needle and drawing the serum from the heart and then stabbing them on the jugular vein so that they bleed to death. And then the serum is packed, filter sterilized, packed, graded, and sent to you for the research. And what kinds of massacre kind of deaths are we looking at for the annual consumption? So if you look globally, we need at least 600,000 liters of FBS, which is routinely being used across R&D labs and manufacturing units and CROs, etc. And we are looking at a 1.8 million deaths of fetuses every single year. And as the demand for research increases, or as the need for the research increases, these numbers will be skyrocketing at a very, very fast pace. And that is very clear by looking at the global serum market. You can see how the growth of serum is happening within the world, from a market value of 1.1 billion dollars to 2.5 billion dollars, having a CAGR of 8.1. There's a massive growth and a massive expansion of serum which is going to happen in the coming years. And serum not only impacts the research, but you can see on the segment wise, it has implication in every segment of research and development, in every segment of product improvisation. So, what becomes something which in our eyes feels very unethical versus compromising the scientific excellence, or trying to compromise the scientific outcomes. How do we maintain this balance between the scientific and the ethical? So, on the ethical, we have a huge concern. Like, we have the animal welfare concerns, where we are killing unborn fetuses, which is very, very cruel. We have transparency issues. When I mean transparency issues, I mean to say that there is so much of variability that comes within the serum composition because of the health of the cow, the calves, the genetic composition, the time of I would say the extraction of the serum, the way the serum is handled, the age. All those things contribute significantly to the variation within the serum lot. And these variations basically, even though attribute a lot to the research outcomes, we somehow tend to neglect that because we have no other alternatives right now available, which can replace a serum. We have religious sensitivities, especially in a country like India, where they consider cow to be, which is given a significance as good as the mother. Doing a brutal, killing of the calves is considered religiously inhuman. So there are religious sentiments which are involved in countries like India, and obviously the public perception of the fact that there is a huge a community of people who are totally against, or who condemn this kind of research, which is based on animal killing, or which is based on animal cruelty. So that's the huge ethical concerns. And on the other hand, we have the scientific concerns, where we are seeing that serum causes a tremendous batch to batch variability issues within the product development process, or within the product life cycle. Serum being so rich in nutrient media and so rich in carbohydrates and lipids, it poses a severe risk of contamination. We've all suffered massively with the mycoplasma contamination, viruses and prions, which can come in from the animal source, or animal viral vectors, which can be transferred through the serum. So they also pose a lot of contamination risks for human beings who are working on serum. Sometimes the serum has undefined composition. What we mean by undefined composition is not that one or two ingredients are missing, but the percentage, or the proportion, or the ratio in which they are distributed from lot to lot causes a significant impact on the cell growth, on the cell proliferation, sometimes even cell adhesion to begin with, on adherent cell lines. Serum also contains a lot of antibodies, because it's coming from the fetus, and which is very rich source of antibodies, so that can cause a lot of immunogenic reactions within the cell culture system. So scientifically also, you can clearly see that there are a lot of issues, there are a lot of challenges which need to be balanced with the ethical concerns. So, looking at this, since on the European Union front or on the Japanese pharmacopoeia front, regulations have already been made very strict and are very loud and clear and impactful on moving into animal-free, research or promoting products which are developed on an animal-free platform or on a chemically defined platform. So India is also moving very fast into the bandwagon and has realized the impact of moving from a serum based to a non-serum based or a non-animal based product development process. We've already been very, very active on the cosmetic front where we've banned animal testing, and this move by the IPC has been something which is very, very promising and hopeful that in the chapter name of 2.7.2, which talks about cell substrates for the productions of vaccines for the human use, it gives a very, very clear, loud, I can say impactful cry that we need to move from an animal derived media to an animal free media. And the recommendations are, very proactive towards the animal free segment, where they are saying that we need to have wherever possible, it clearly mentions wherever possible, please use animal free media for the productions of human vaccines. It also very clearly talks about the validation data, because a lot of the products which are already available within the market, which are in the vaccine segment, already have validated data. So moving from a serum based to a serum free media would mean complete validation for the product, and it clearly makes it mandatory and says that comparable data is must. On the regulatory front, it also gives you the importance of aligning with the WHO and the EMA guidelines, and requests that these guidelines be followed strictly for the development of the products. And on the R&D front, it also encourages using serum-free systems for development of newer products, which are going to come into the regulatory chain for approvals. So, having said that, when we have a strong commission which is proposing the use of animal-free products or developing animal-free products, we also need to very clearly understand that this paradigm shift would mean a lot of challenges on the scientific as well as the regulatory front, because no matter how fast the science is progressing, there are certain strict regulatory protocols which need to be abided by, and whether the regulatory departments or the regulatory authorities are flexible enough to incorporate these changes in the transitioning phase. So, looking at the type of challenges, we see that adaptation is one of the biggest challenges which the scientific and the regulatory community is going to really, really face on moving to an animal-free media. On the scientific front, it is obviously the fact that cell lines which have been cultured or conditioned or groomed for serum-based production of vaccines need to move from a serum-based to a serum-free media. So that transition is going to be a scientific challenge. On the regulatory front, obviously, it would mean resubmissions of the paperwork, the documentations, the approvals, which would mean having a more stricter defined guidelines for product approvals. On the media front, every product that needs to be developed would need a customization. So we can't have a one dress that fits all kind of attitude over here. So every product which is now going to be moving on from serum to serum free media would need a lot of customization depending on the kind of product that is being developed or the application for which it is being developed and the scale at which it is being developed. That also is going to have a lot of significant impact. And regulatory front, obviously, the regulators will look for GMP compliances for the productions of such kind of customized serum-based medias, and they are being chemically defined and having the approvals or the right kind of ingredients for the development of the product. So that also going to play a very, very significant role. The process changes obviously is going to be something which is going to be a big question mark from an industrial commercialization point of view, because that is where the industries would really have to invest a lot of money in changing the whole portfolio of process optimization from a serum based product to a non serum based product or an animal free product, and that is going to require a lot of time. It could also mean changing a lot of protocols and development of new SOPs, which will be like cost intensive, time intensive activity for the industries, especially from the commercial production point of view. On the regulatory front, these are going to have a lot of significant impact, because it could also mean redoing the whole process of approvals, which could again have be like a heavy on the pocket expense kind of for the industries. Other issues would obviously mean having supplementation in terms of engineering for better development process. And most important on the regulatory front would be an international harmonization, which is one of the biggest challenges that we are facing right now, that there is no uniform code for such kind of products coming into the market. And this uniform code will be something which is going to be very important when it comes to marketability of products which are being developed on an animal free media. So if these products have to be universally accepted and have to have the global market for their sale, then there has to be a lot of harmonization which needs to happen across all kinds of regulatory bodies across countries, so that it gives you the flexibility and the freedom to trade products. So, how well is the regulatory right now gamed for accepting serum-free media? So, I think they have certain key guidelines, and if you are able to fulfill these norms, you are in the game of, the product development on a serum-free media. So, they're looking for composition transparency, means they are expecting you to completely disclose the synthetic media or the chemically defined media that you're using for the production of these processes. Obviously, they are focusing on absence of animal derived component. When we mean absence, we are not saying complete elimination, but it also could mean reduction in the usage or in the utility of animal derived components. What they are really looking at is the batch to batch consistency, because since there is no natural product being involved here, which is going to be a variable factor, which is going to be a factor which is going to impact the outcomes of your research, they believe that moving on to an animal free or a serum free media would give you a better batch to batch consistency in terms of products, which could make data very reproducible. Obviously, no harmful agents, having the GMP compliance, validation studies, cross compatibility studies, all of this are a part of their routine mandate. So, coming to how HiMedia is, playing a very significant role within this segment is HiMedia. Let me give you a quick introduction. It's a 50-year-old company in India. It's an Indian indigenous company run by a close knit family, and the company is a pioneer in molecular biology and microbiology, and has now set up a very strong and a very potent cell biology division, which is actively working on having animal friendly products being pushed into the market. And we have this very beautiful product called as new Sera, which is a serum replacement solution that has been developed in-house, trademarked by us. So what is this basic product? So it's basically a serum replacement solution. Can be used for both adherent as well as suspension cell lines. And since it is chemically defined, it will ensure that there is batch-to-batch consistency, reproducibility. And the beauty is that since it's a serum replacement solution, the adaptability quotient between the cell line is very, very high. It's very, very good. And the cherry on the cake is the fact that using Nucera is actually going to give you a 30% cost reduction in your R&D workflow, which is going to be like a win-win situation in both the cases of moving from an animal dependent to an animal free media, plus having the cost benefit advantage. And obviously, knowing the fact that it's a substitute, it is going to promote more ethical research. So, why would we want Nucera? Because Nucera, one bottle of Nucera can save two fetuses, which are being killed for serum. So, you can imagine the kind of contributions it is going to make in the uh, animal prevention sphere. Like, using this can have a significant reduction in the number of deaths that we see happening very, very, very rapidly. It has low protein content, so less interference for any DSP process where you're looking at high cell density expression of proteins, or you're looking at production of MABs, where there's a lot of host cell protein interaction that happens, or a lot of carryover protein contamination that comes in from the media that is being used. Since the concentration of proteins are very low, it gives you a faster turnaround time for purification of proteins. Lot to lot consistency, since it's chemically defined, and obviously it is going to be compatible with all kinds of cell culture media, and it is free of any biological variability or any biological entity that comes in from the host. It needs no screening and no batch reservations whatsoever for its activity. So here's some data that we would like to share with you, on the validation of FB from Korea. So we have this very, very significant partner called as Dr. Oh Park. He's the deputy director of the Vaccine Innovation Center in the Korean University. And he basically decided to take up the study for validation of our new Sera versus the conventional FBS, which is Serena, which was being used in their lab. And what he did was a comparative analysis across all cell lines, and showed that Nucera, in especially MDCK cell lines and Vero cell lines, which have been actively being used for their vaccine development, has better performance outcomes as compared to the standard FBS. In the other cell lines as well, you can very clearly see the representations that either it is at par or a shade better than the conventional FBS. Moving on for the study, what was really important was, okay, this is not, okay, so they basically conducted a lot of cell morphology studies on MDCK cells across multiple passages, right up to 12 passage, and they saw that there was no difference within the cell morphology on a standard FBS versus new Sera across 12 passages. So they had a very stable cell morphology. They also did it on Vero, and Vero cell lines also showed no difference in cell morphology right up to 12 passages. They went on to understand the cell growth and viability, and did a complete cell number analysis, and saw that at higher hours, the new Sera was performing much, much better than the standard conventional FPS. So there was no difference in the cell growth and viability. If you look at the viability data also, up to 72 hours, we see that the cell viability is as compared to standard FPS media. So going for the plaque assay, which is one very significant assay for the development of vaccines, you can see that almost up to 13 passages, there is no change in the plaque formation or in terms of the morphology of the plaques, both on an FPS as well as the new media for MDCK cell lines. And for Vero, there was something which was very, very significant that we observed, that there was no difference in the plaque morphology and the titers between FBS and new but on the long-term subculturing, we saw that FBS was having a cell loss, whereas new was actually being, there was a lot of having a very high cell density. So there was a lot of cell shredding which was happening at higher time points on an FBS, which was not observed in the new So they kind of were more stabilized in a new supplemented media. The same goes for the titer values, which were seen across multiple passage levels. And they found out that there was no significant difference between the titer values of antibodies obtained from FBS as well as new across multiple passages. And nuclear staining also confirmed that there's absolute intact cell morphology between MDCK as well as cell lines. And in terms of biological activity, we saw a wound healing assay was done for MDCK and Vero, both in the presence of FBS as well as new and they found that the wound healing activity was identical in both new and FBS. That means biological activities of the cell lines are also absolutely intact and preserved even without FBS. So I think that significantly proves quite a lot of information on the front that these products are a classical example of that there are alternatives available which can replace a serum. It's only about education. It is only about validation. It is only about trying multiple opportunities and then finding out what suits best for your research needs. Other than New which is one of our very salient featured products, we have something called as a high wedge media, which is a plant-based media full of peptones, hydrolysates, and it basically, because it's a plant-based media, there is no contamination, no issues of viruses and all, and it is a media which is being actively used in our cultivated meat segment. So a lot of people are buying this media from us who are working on the cultivated meat platform, where they're looking at enhanced growth from their fish cell lines. So they're using this kind of medias, which is offering better alternatives. We have a smart media, which is basically a xeno-free systems for culturing the human T cells. Again, it has been optimized for R&D purpose, basically to make sure that we have a serum-free environment for T cell proliferations. We have a stem-in-one media, which is a defined media for the mesenchymal stem cell proliferations. We have a cell-in-one media, which is again a serum-free media for cultivation of marrow, PK15, MDCK, MDBK cell lines, basically very, very active cell lines which are focused towards vaccine development and vaccine production. We have an HK1, which is basically a media that was optimized for the COVID-19 vaccine productions. We've been very, very bullish on this product, and we've been able to supply to a lot of companies which are working on the vaccine front. We have a BHK-in-1 media, which is again formulated for the foot and mouth diseases. We have a very classic example of BioVet, which is a vaccine development company based out of India, which is focusing on animal foot and mouth disease vaccine, who's using our media for the production of their for the production of these vaccines. And then we have the enzyme reagents. Obviously, we have a lot of, trypsins and other kind of products which are not having any trace of any enzyme animal-based products within them. So coming to the conclusion part of it, what I basically want to talk about is that we all need to focus on embracing a sustainable and an ethical future within our cell culture segment. My information, the point of sharing this information is to basically let you know that there are ways by which we can avoid serum within the media, and we can get better or at least at par results and systems which can give us the desired outcomes. So the need is very clear. We need to move on from the traditional serum-based media to a non-serum based media, which basically raises a lot of ethical concerns, scientific concerns, reproducibility concerns, and many more. Having an animal-free media is going to be the future, because it is going to align with the 3 hours of replace, reduce, and refine principles, which is I think one of the most talked about principles in non-animal based segment. What it does, it reduces the variability and the risk of contamination, which is one of the major reasons why there is a need to shift to an animal-free media. It obviously gives you a better prospects for giving you data which is more standardized, more reproducible, which can be validated across many countries to give you more comprehensive or a more holistic kind of results. India has the opportunity right now because we are on the forefront of the innovation. We are having our government policies and regulations and the committees like the IPCs, which are strongly promoting this concept of moving into the animal-free space. And obviously, as scientists, it is our commitment to make sure that we are coming up with more innovative, reliable, cost-effective, and regulatory compliant products and solutions in the segment so that we can offer better outcomes in terms of research or product development. So I think, Ankita, how much time do I have more?
I think enough that you can conclude maybe a minute.
Yeah, so I just wanted to kind of give you the overview that we have a lot of happy customers who have given us very strong testimonials on Newsera, on our STEM-in-1 product, on our HEK1 product, on our BHK1 products, which have actually created them, given them an edge in terms of reducing the lead time for purification and getting better outcomes, economical outcomes within their product development. So it is not a science, or it is not a concept that we are talking only in papers, but at HiMedia we have shown the practical applicability, the commercial viability, the need, and the positivity of acceptance from the scientific community to show that there is a scope, there is future of moving into this animal free segment. And we continue our zest in promoting the animal free segment very profoundly within HiMedia. We are already in a product pipeline of new products being developed. We are into the customization of media, which would require any support from the clients who are looking at developing animal free media. We are here, we can offer you our services on that fragment, and help you with commercial productions as well. So I take pride in letting you all know that HiMedia is an indigenous manufacturing company having 18 manufacturing units within the country. And we have one of the largest manufacturing units in the whole world for solid and liquid media. We have an average capacity of 60 tons per day for production of solid as well as liquid media. And we are open to all collaborators who would like to collaborate with us in developing science and promoting research and developing products, commercializing products, scaling up products which are going to be on an animal friendly platform. We have that commitment. You can see that our logo itself says life is precious. So be it a human or an animal, it means a lot at HiMedia. So I take this opportunity to thank a lot of people who've been the backbone for today's webinar. My first heartfelt thanks to Dr. Ankita for being so proactive and giving me an opportunity to interact with her and promote HiMedia on a very global platform like PETA International. I would also like to thank the Vaccine Innovation Center, Dr. O, for being so voluntarily coming up to help us to validate our studies at the Korea University. The business team at HiMedia and the research team at HiMedia, which has been very, very active in developing this product. The management team at HiMedia, my directors who've been so warm and have given me the free hand and the opportunity to interact with all of you. The tech and the IT team, both at PETA India as well as at HiMedia, who's been responsible for designing the slides, designing and creating the content, the data, and has been very, very instrumental in helping me deliver this lecture today. So that brings me to the end of my presentation. Thank you so much, everyone, for giving me a patient listening. And I will be more than happy to take up any questions that are there in the forum. Ankita, you will have to guide me if there are any questions. I think there are two questions.
Yeah, we'll take all the questions at the end of presentations, like after Kamalnayan's presentation. Yeah.
Sure. Yeah. So, yeah. So thank you so much, Ankita. If there's anything else, I'll be more than happy to help. Thanks.
Thank you so much, Dr. Pratiksha, for such an insightful presentation. Moving on to our next speaker for today, that's Mr. Kamalnayan Tibrewal from Biokraft Foods. Mr. Kamalnayan is the founder and CEO of Biokraft Foods, a pioneering food tech startup at the forefront of cultivated meat innovation in India. Mr. Kamalnayan pursued BTech in biotechnology from Maharishi Markandeshwar University, Ambala, and MTech in Green Technology with a research specialization in cultivated meat and 3D bioprinting from Institute of Chemical Technology, Mumbai. Under his leadership, Biokraft Foods aims to bring India's first cultivated chicken meat using advanced 3D bioprinting technology, offering a cruelty-free and environmentally conscious alternative to conventional meat. He actively collaborates with research institutions, investors, and industry partners to accelerate product development and market readiness. Mr. Kamalnayan's work has positioned Biokraft as a key player in the alternative protein space, earning recognition across national and international platforms. It is now my pleasure to turn the floor over to Mr. Tibrewal.
Thanks, Dr. Angita, for the warm introduction, and thanks for this opportunity to present our work at the PETA. The presentation given by Dr. Pratiksha as well. I mean, it was very great, setting us the platform for the required insight on the FBS, and very glad to share the insights where FBS is lying now in the cultivated meat industry, and just walking around where we are and how we are trying to minimize this base use in this whole scenario. So yeah. So good evening, everyone. I'm Kamalnayan Tibrewal, founder of Biokraft Foods. So it's an honor to be here to speak on a topic that goes beyond science, and it's about ethics, sustainability, and our collective future. Today, I will walk you through our journey towards creating real meat without compromise. Cultivated products that's not only animal-free at the plate, but also in the process. So let's begin with the basics, where what is cultivated meat. Cultivated meat, as we know, is produced by growing cells directly without raising or slaughtering the animals. This radically new way of making meat directly addresses three global issues. Environmental impact, like cultivated meat reduces greenhouse gas emissions, land use, and water consumption drastically. Animal welfare, no need for animal farming or slaughter, and food security, where it offers a resilient, disease resistance, and scalable alternative in a volatile global food system. So it's a convergence of science and values, and it's happening right now. So, our solution is cultivated chicken meat grown directly from animal cells without slaughter. We use proprietary 3D bioprinting technology, non-GMO, antibiotic-free ingredients, and grass material to create a meat in a very controlled environment. The result, contaminant-free, nutritious meat with significantly lower carbon emissions. And it's not just a product. We have filed a complete process patent and developed our own in-house bioing to make it more scalable and economical. So this isn't just about meat. It's a sustainable nutrition engineered with ethics at its core. So step by step, if you see the cultivation process. So let me just briefly explain you like what's the step-by-step approach. First, start with the cell sourcing. That we start with a high quality immortalized chicken fibroblast cell line. These are ethically derived and can grow indefinitely, so no repeated animal use. Cell cultivation, the cells are expanded using a defined media, in sterile condition to reach high densities that's ideal for a food application. Scaffolding, we blend the cells with a proprietary algal and plant-based bioing that serves both as a scaffold and as a nutritional base. And 3D bioprinting, the bioing that's made is finally fitted into a 3D printer and printed into the shapes like chicken breast that's ready for cooking and no post integration required. So it's a seamless integration of biology and food technology. So let's talk about FBS, the fetal bovine serum, and like what's the whole story is about. So traditionally used in cell culture, it comes from the unborn calves, like Dr. Pratiksha told, and that alone makes it very unacceptable in a process that aims to be cruelty-free in the first place. But going FBS free is more than an ethical choice, but it's also about setting new industry standards, pushing sustainability goals, and creating a fully animal-free product from start to finish. If cultivated meat is to fulfill its promise, eliminating FBS is non-negotiable. Uh, but there's this FBS dilemma where there are different challenges and comparisons. Let's take a moment to unpack one of the biggest hurdles in cultivated meat production: replacing FBS with something that's animal-free, ethical, and scalable. On the left side of this table, you will see the characteristic of FBS-based media, which has been the industry standards for decades in cell culture. On the right hand is where we are headed: serum-free media, or SFM. The first and most obvious difference is the source. FBS is animal-derived, collected in a process that is inherently unethical. In contrast, SFM is made of defined ingredients, plant-based, recombinant, or synthetic, which gives us a cruelty-free path, very forward in this. But ethics isn't the only driver here. Cost-wise, FBS may look cheaper upfront, but it fluctuates widely depending on availability. And as demand grows, so does the price volatility. On the other hand, SFM is more expensive today, especially because of recombinant protein, but it offers a long-term predictability and scalability. One big advantage of SFM is consistency. With the FBS, every batch is different, like cooking with a mystery of spice mix. But SFM gives us exact repeatability, which is essential when you're scaling up the production. From a regulatory standpoint, SFM also simplifies things. Regulators are rightly cautious about approving food grown in animal-derived undefined groups. So SFM clears that hurdles. And finally, scalability, where FBS is tied to a slaughterhouse output. It doesn't scale with demand. SFM, by contrast, can be produced in bioreactors just like the cell stem cells. So that's future-proof solution. So while switching to SFM comes with its own R&D and cost challenges, it's clear that if cultivated meat is to succeed at global scale, this is the part what we need to take in future. And when we started reducing our serum in our media, we quickly realized this isn't something you can just switch off overnight. Cells that have been spoiled by the richness of FBS doesn't simply adapt to a minimalist environment. You have to train them gradually. We used a method called sequential adaptation. That means we start by culturing cells in the regular high serum media and then slowly reduce this FBS concentration over multiple passages, step by step, while introducing serum-free components in parallel. Think of it like a winning. If you remove serum too fast, the cells either stop growing or drastically change their behavior. Now, why this take time? So, because every small adjustment impacts cell morphology, doubling time, and viability. We would reduce serum, the cells would survive, but grow slower. Then we would tweak the media composition and supplement it with some specific growth factors or nutrients and test again. Each iteration took days, sometimes weeks. So we evaluated, over multiple serums, like serum-free formulations, being specific, more like five, around more than five. But some supported morphology, but not proliferation. Some gave decent growth, but failed in suspension condition, which are essential for scale-up. Another challenge we faced was nutrient deficiency. Many SFMs on the market, especially those designed for pharma, had 35 to 40% lower levels of key amino acid, vitamins, or trace elements compared to FBS. This meant our cells were metabolically starved, and we had to rework the nutrient ratios. So today, while we have significantly reduced our reliance on FBS, like down to around 1% with 5% SFM in some protocol, we still incorporate a minimal amount as we continue optimizing. So this isn't being stuck. It's about being methodical. A cell line that grows fast but loses structure or nutritional value with time isn't a win. And that's why this journey has taken time, because we are not just growing cells, we are building food. And food, unlike cell lines, has to deliver on safety, taste, texture, and scalability all at once. So today, while we have significantly reduced our reliance on FBS down to about 1% with 5% SFM with in some protocols, we still incorporate a minimal amount as we continue optimize. This isn't about being stuck. it's again, as I said, it's about being methodical. And like I mentioned, that this journey, will take some time because it's just not about growing scales, cells. And we are building food, and unlike cells, food is dependent on several parameters. So we will eventually get there, but yeah, it's taking some time. We have successfully achieved on 80% reduction in FBS use, which means our cells are ready, already growing in significantly lower serum concentration than they started with. But what's even more important is that uh, you know just not for the sake of the metrics, it's functional aspect as well. Our cells maintain good morphology, viability, and are adapting well to suspension conditions, which is essential for scalable production. Now, are we 100% FBS yet? No, not yet. But we are very close. We are still using a small fraction of serum because some critical growth factors, particularly those that impact proliferation rate, are hard to replicate fully. But our commitment is absolute, and we see FBS, free as not as just as a checkbox, but as a threshold that entire industry must cross. And we are investing heavily in reaching there, not just in the lab, but in a way that works at both at commercial scale as well. So, I mean, why it's hard? I mean, technically, the real challenge isn't removing FBS; it's understanding it well enough to replicate its effect. And even when something is working in lab flask, it often breaks down at scale. Cells that grow beautifully in a 10 ml may behave very differently in a 100-liter bioreactor. So this is why FBS free is not just about switching ingredients. It's about solving a biological equation, which what might still fully don't understand. Thankfully, but we are not alone on this part. Multiple alternatives have been explored, and like plant-based hydrolysis provide affordable amino acid and peptides. They are recombinant proteins that's giving us high purity growth factors, but at higher cost. But algae extracts are showing promising results in improving proliferation. But at Biokraft, we are combining these two to create a serum-free formula that's both ethical and scalable. The future lies in stacking this innovation, not relying on just one. And of course, it's not all smooth sailing. Many of these recombinant inputs are still expensive. Media still requires custom optimization for each cell type. It's like, not one size that fits all kind of thing. And many SFMs were built for pharma, not actually for food. So this is where we need collective effort from startups, from researchers and policymakers to reduce cost, increase access, and make serum free the new normal. Again, one of our strongest belief at Biokraft is that no one company can solve this alone. We need to foster shared learnings, open access to cross-sector insights, whether it's from biopharma, fermentation, or agri-tech, and align on a supportive policy frameworks. This is not just a technological challenge; it's a system challenge, and solving it requires us to move faster, not in silos. So, looking ahead, here is what we are focused on: finalizing a fully FBS-free formulation for use in bioreactors, driving cost reduction by sourcing scalable food-grade inputs, working closely with regulators to ensure compliance and readiness for market entry, and expanding our portfolio from ground meat to structure cut and eventually in adjacent sectors as well. We have already gone into cultivated seafood as well. So we see it's just not that we are a food company, we are a tech company as well, and so we have to solve a problem that's a fit for all. And so all this take time, but every decision is guided by the same principle that real meat, no compromise. And by developing truly a base-free cultivated meat, we don't just create a product, we help redefine the global food ecosystem. We are reducing pressure on the planet, ending hidden cruelty, and increase resilience on how we feed billions. So this is a moral responsibility, and it's one that we at Biokraft are proud to be leading on. And at Biokraft, our vision is simple: that real meat, no slaughter, no compromise. That's not just a tagline; it's our roadmap, like our identity and our contribution to a better world. We are incredibly close to delivering on these promises, and I invite all of you, like as a scientist, entrepreneurs, regulators, and advocates, to be a part of this change. And thank you so much for your time and attention. And I'm very happy to take questions or discuss our progress in more details. Thank you very much.
Thank you, Mr. Kamalnayan, for your informative presentation. Moving on, we have some questions that I can see from the audience, and we'll try and answer as many as possible in the next 15 minutes or so. Pratiksha, there's the first question for you. So could you please elaborate on what you mean by performance of new Sera is equivalent to that of 10% FPS? And also, is new Sera media completely animal-free?
Okay, so I'll take the first question. When I mean that the performance of new Sera is in comparison to 10% FBS, we have a lot of performance parameters which play a significant role. So, what we've basically looked at is the cell morphology and the proliferation rate. We've worked on, since we were working on the vaccine cell lines on Vero, we also looked at the titer values, the plaque assay activity profile, and when we do so basically it was a physical, metabolic, and a proliferative comparison portfolio which we wanted to develop for the product, and then understand the implications of with FBS, without FBS, how the cells were performing. No, I can't say without FBS, but with FBS with substitute is what we've worked on, and we found that the substitute new Sera is doing a much better performance, or at least at par performance in certain cases, for the various parameters which were under question. So on the titer values, we saw much better results as compared to FBS. Whereas on the others, like the proliferation or the, on bioassay, that is wound healing, we saw as good equivalent performance in presence of FPS. Now, coming to the second question, which was, can you just...
Yeah, the question was whether a new Sera is completely animal-free.
So new Sera is, I can't say it is completely animal free. It is having only 5% animal component into it, which is basically in our new product development, we are trying to eliminate that 5%, animal composition, which is there in the new Sera. So it is 95% chemically defined, 5% animal component is there. So we are almost close to achieving a completely animal-free product. So our R&D team is very strongly working on replacing that 5% and making a completely chemically defined media, which can be like 100% replacement to serum.
Okay. And what would the cost of around 250 ml of new Sera be?
The new Sera is 30% cheaper as compared to FBS. So if you consider a 500 ml bottle, which is a standard FBS bottle, it is 30% cheaper. I would say, I don't want to name companies, but if you take the market players and average out a standard FBS bottle for 500 ml, we'll be 30% cheaper.
Right. Thank you. So the next question is for Kamalnayan. So you showed, the cell morphology and doubling time data, but is there any other sort of validation studies, maybe morphological analysis, cell functionality parameters that you've looked into while transitioning your cells with like reduced reliance on FPS?
No, thanks for that, Ankita. So, while we are doing so, first and foremost is that, what we have to look into is that while we are transitioning, the cells are stable in nature as well. And the stability is just not on the morphological, but also in the genetic level as well. That whatever recombinant proteins or whatever SFM being we are using, it's not affecting cells at, on the genetic level as well. So that's one of the important aspects that we have to do. I mean, we have already done that. Some safety data evaluations like AIM study, chromosome elaboration test, this kind of test. Ultimately, whatever you do, even you have achieved everything, ultimately your cells has to pass through this safety evaluation test. So that is something that our product stands on, and that's how we have already done it. But while you're doing while you're doing this transition, I mean the visible parameters that usually is seen is the proliferation and the morphology that usually you opt out, because any change in that is not acceptable anyway in the long ratio, because if anything is impacting your proliferation and your doubling time is getting increased, so that means your overall batch productivity is getting decreased. because the more the time the cells will take to replicate, or the more the time it will take for higher density, anyway, it won't help you. So the challenge would be to, these two parameters are the basic for easier understanding that, while you're transitioning, just, focus on this while you're evaluating even others as well. I mean, this is something that we really focused on.
And there's also a following question that not just FPS, but what about enzymes like trypsin that are also animal derived? How are they being replaced in cultivated meat production?
So once your cells are in the suspension, culture, you anyway don't need trypsin anymore. While your cells are in adherent mode, I mean, when you're just culturing in the FBS, so FBS inherently contains adherent factors, and that's one of the problem why you can't really go from adherent to suspension just while using like the 10% FBS what is standard. While you're transitioning to SFM as well, you are again we are modifying that, and you are not putting adherent factors into that. So that from adherent to suspension when you're going, so again you don't need to worry about trypsinization. Again, that's a cost saving step. What I will tell while you're moving to transition, the cost of trypsin is also getting saved.
Dr. Pratiksha, do you have anything to add to that?
Yeah, I mean, to that, I would say that, okay, I think what Kamalnayan made a point was very clear that, anyways, for the commercialization or for commercial development, you will never be going on an adherent cell-based system. It will be more on a suspension cell-based system, bioreactor-based systems for scaling up the product. So that's where trypsin really doesn't play a very significant role. But even if it does, we do have recombinant trypsin, which is a very, very easily available in the market, and it is at a very throwaway price, so as compared to animal derived trypsin. So that should not be a problem. We do have cell-based recombinant trypsin, chymotrypsin, both being available commercially in the market, so it should not be much of a concern.
Pratiksha, there's another question. What are the key challenges in scaling up FPS-free media for commercial biotech or cell-based applications?
So commercial challenges, I don't see any as of now, because what you do on a commercial level is basically a stepwise conditioning and scaling up. Now, when you are already culturing at the pilot level, your batch for a serum-free media for production of say antibiotics, or production of monoclonals, or production of any lead vaccine molecules, scaling up should not be a very, very challenging task. Nonetheless, I cannot say that you can compare apples to apples and mangoes to mangoes. It's not going to be a very systematic scale up. There will be a lot of bioprocess challenges which may come in. But the best part about scaling up is that, since this is a very chemically defined media with very low protein concentrations, your downstream processing for any product development becomes a very, very affordable task. The purification of the products is faster, is cheaper. So you will be able to develop products which are more economical if you are going on a serum-free platform. Plus, because you are using a serum-free platform and there is a cost reduction happening at the product development stage itself, there's going to be an added advantage that you will be able to bring down the total product cost significantly or drastically at the end of the process chain, because you are reducing FBS, so there's no cost intensification that is happening over there. Plus, your purification steps become very limited, so again there is a lot of cost saving that happens over there. So scaling up actually will be a very commercially viable activity, which will be on a serum-free platform. But yes, we really don't know, because we still don't have a single commercial product in the market which is developed on the serum-free platform. So we don't know the biochemical or technical challenges that we may face coming in the future. But I believe that if we've got success at the pilot level, scaling up should not be a big task.
That's interesting to know. So I can see a lot of questions pouring in. Pratiksha, there's another technical question for you. What is the percentage of cells getting attached post subculture in serum-free formulation?
So I think on the percentage of cells, you can, I've shown the data, there's 100% attachment, but the confluency is close to 80%. Even after 12 passages, we've gone up to almost 18 passages, and we've not seen any loss of adherence within the cells without in the absence of serum. So I think you can consider that the attachment is almost 100%. There's no issue on that front.
Okay, thank you. Kamalnayan, what components did you identify as critical to replacing FPS, and how did you source or engineer them?
So that's a very interesting question. I mean, technically, we are not optimizing SFM at our level, but we are working with our collaborators. But there's a catch in that. Although we are an end product company, still we have to participate in this whole activity because, again, as I mentioned, there is somewhere a gap between the understanding the chemistry between the fully, what was present in the FPS and what is required by a company. So when we are working with the companies and when we're optimizing and we're telling, as I told that there is still a 35 to 40% lack of concentration between what's generally present in FPS and what's coming in SFM. So I mean, from a company perspective, who are providing this SFMs, the real struggle is that some companies are trying to match all the components while maintaining that cost, because again you have to match the cost as well while maintaining all the components. But the issue is the level of the concentration of these components, because some concentration is so minimal that sometimes it's not well enough or in a level to feed the cells. Now, again, this was not a one solution for all. Again, we have to curate it according to ourselves. So now we have to again, every time give feedback to our collaborators. So this is the thing that needs to be added, and mostly it's happening on the growth factors level. Very honestly, where you different EGF, HGF, and this kind of growth factors. Usually we have to optimize this kind of thing. So apart from focusing on our end product, which is our forte, we have to work with SFM companies to even because again, if we don't do that, again we won't be able to go to our end product at that kind of level and scale, because again we have to do the production, right? So it's something that it's a continuous loop that's happening. But I think understanding the real chemistry of when we are talking about substituting FDS is what the real challenge is. I think that's what we really need to work on.
And one of the questions is if you checked the proteome profile of the cells when they were grown serum-free.
So we don't we haven't checked the proteome of the cells, because we test the final protein analysis or anything of the final product, because we are talking about final product here. The cells are going in the final product. So whatever protein analysis we do or whatever amino acid analysis we do it for the final product. Even for the safety data studies we talk about, another thing is the final product we are talking about. So we go with that.
Okay. There's a general question. I'm wondering which one of you would like to answer this. Is there any plant source for cell culture media? If yes, which one is the best? And what are the challenges in developing a plant-based cell culture media? If either of you has experience in developing such a media.
Yeah, so HiMedia has a high-veg product, which is a plant-based protein hydrolysate, which is used for cultivation of, which is used in basically for developing media for cultivation of cultivated meat. So we've seen that it has enhanced the growth of the fish cells, which are totally focused towards cultivated meat segment. The challenge I would think about what I'm not very confident on that, because that's one segment which I've not worked with. It's more to do with the plant tissue culture segment. So they are the ones. It's a very different vertical which is working in that segment. But I believe, to the best of my knowledge, would be the solubility with these kind of protein hydrolysates coming from the plant source. So they basically are a very, very a complex molecule with solubilities being very minimum. So I think the biggest challenge would be to have a completely soluble product, which is which will have homogeneity in terms of media. But I believe that with high-veg, we were able to solve that problem, and it is being very well commercially used in the production of cultivated meat.
So Pratiksha, for you again, are there any presence of growth factors used in the preparation of new sera? And if you are using recombinant ones, did it affect the final cost of the product in the market?
Yes, there are growth factors because they are the key essential ingredients which are required for the for serum's performance in a cell culture media. We are using the recombinant growth factors which are commercially available. Most of these growth factors have been developed in-house by us, by our clones. The composition is a proprietary composition, which I cannot disclose to you over here on this platform. But yes, growth factors are an essential part of the serum-free media development, and we do use recombinant growth factors. And if we come to the cost comparison part of it, the concentration at which they are being used in these products is so minuscule. They are used in nanograms and micrograms. Frankly speaking, it really doesn't impact the overall cost of production. And when you're looking at small scale production, it really doesn't make that a very significant impact. As you can see that new Sera is 30% cheaper than FBS. There's a fact that the recombinant proteins are being used in a very, very minuscule and I would say a significant, but quantitatively not significant manner. So it really doesn't impact the overall product outcome as well as the cost.
Great, thank you. Kamalnayan, how do you ensure the genetic stability and consistent proliferation across multiple passages?
Yes, so for that we have done the STR analysis and DNA sequencing of different passages. Like talk about like one at when it was in at 154, at 160, and then when it was in suspension. And when we compared this particular cell line in the whole cases, so there was no variance particularly present in this different forms of the DNA at different passages. So that's one way where you do the STR analysis and you compare or do the whole genome sequencing and compare everything at the genetic level. While you're ensuring majorly going through the references of other doses where you have to focus specifically on anything mutation causing genes particularly but overall the cell line that we're using and when we have done this analysis there is no variance and the cell line is completely stable in that form. So that's one way of ensuring. And again going back then the final product that we talk about, you do the safety data evaluation, the Ames test, chromosomal aberration, micronucleus test. So these are the tests that ensure that even if you don't, there's one thing that we have seen that even if you don't do things specifically on the cell line or other things, again, going back the final product as well. Again, the final product is something that's been getting consumed. So you have to ensure that the safety data test, whatever you're doing at the final product, that should pass or that should be compliant. And anyway, that already indicates if there's any mutagenic components or not.
I see there are a lot of people who are already excited to try your product. So they're asking if your product is already available in the market now.
So we have done a couple of market validation and they're doing external market validation as well. We are, I think, if you go on our website on experience our product column, so we have maintained a wish list for that, and people can really sign up, and we are very happy to host people at different occasions. That's something. But yeah, I mean, unless it's approved by regulatory, you have to wait a little in that case. But I'm hoping in early 2026 because we are getting our dossier filed by next month. So yeah.
Great, thank you.
All the best.
I just see one last question. So is there any database to search for companies that are offering serum-free or animal-free chemically defined media?
I don't know about database, but definitely PETA's website can give you a lot of information on that front. They have a whole list of people who are in this business, so who are actively doing research and have commercially launched products available. So I think you don't need a database. You have enough of open source information in the public domain, which will give you that kind of an information.
So we do actually have a web page where we've shared a lot of information about FBS, how to make the transition, and there's like a list of companies that are offering serum-free and chemical-defined media. Also, for those interested, there's also a fetal calf serum-free database available online that basically gives a very comprehensive overview of the commercially available serum-free medium and also other alternative medium compositions that have been prepared in-house. And there's like a lot of scientific literature published on those studies. So that's a very interesting website if anybody is interested in checking that out.
You should visit that page.
Yeah. So I don't see any more questions. So I think I think this was a very helpful question and answer session. Thank you both for sharing your valuable insights. Since we have no more questions from the audience, I think we can conclude the webinar for today. So thank you again, everyone, for joining today's webinar, and to both of our speakers. If you guys have any questions or comments about the webinar or for any of the speakers, please do feel free to write to us at the email ID on the slide, and just enjoy the rest of your day. Thank you so much.
Thank you so much, Ankita. Thank you so much, everyone. Kamalnayan, it was nice meeting you. We'll be in touch.
Yeah. Thanks, everyone. Thanks, Ankita.
Thank you. Thank you.
Bye.